Cutting a crown-of-thorns starfish in half does not kill it. It makes two. In 1969, fishermen and recreational divers along the Great Barrier Reef started doing exactly that. Armed with knives and bare determination, they waded into outbreaks and sliced thousands of animals, cut apart, scattered across the reef floor. The water smelled of brine and severed tissue. They went home believing they had helped. The population grew. Robert and Dean at the University of Queensland had been watching this unfold since the first documented outbreak hit Green Island in 1962. Dense aggregations of Acanthaster planci, the crown-of-thorns, moved across the reef like a slow tide, each animal extruding its stomach directly onto living coral and dissolving the tissue beneath it. One animal consumed 5 to 6 square meters of coral per year, according to Peter Moran at the Australian Institute of Marine Science. By 1979, a second outbreak wave was rolling across the reef system. A third followed in 1993. Each one worse than the last. Then came a discovery nobody expected. The eradication attempts weren't just failing. They were feeding the problem. In 1973, Dean and his colleague W. Stablum published a direct warning. Their words were precise. The practice of chopping up crown-of-thorns starfish should be discontinued, as it may result in an increase rather than a decrease in numbers. But nobody stopped. By 1985, the Australian Institute of Marine Science launched systematic monitoring. Transect lines, clipboards, divers counting animals in columns. The numbers in those logbooks climbed sharply through the late 80s, then through the 90s, then again after 2010. The fourth outbreak wave, the most severe on record. The question that nobody had fully answered was mechanical. Not why the outbreaks happened, but why removal made them worse. What precisely, inside a severed arm, was capable of producing an entirely new animal? That answer exists. It involves a fragment of tissue no larger than a coin, and what Dione Dika confirmed in a University of Sydney laboratory in 2020 will change how you understand the word eradication entirely. The solution seemed obvious. Send divers. Remove the animals. Repeat until the numbers drop. By the late 1960s, the Great Barrier Reef Marine Park Authority and the Australian Institute of Marine Science had coordinated exactly that. Funded removal programs, trained teams, dive rotations across the reef. The logic was clean. Population too high, reduce it manually. Simple arithmetic. Except the numbers went the wrong way. Field reports from culling operations between 1968 and 1971 documented something nobody had predicted. Surveyed sites that had been cleared showed higher crown-of-thorns counts in follow-up assessments. Not stable. Higher. The divers were working harder, logging more hours, pulling more animals, and the reef was getting worse. Robert and Dean at the University of Queensland spent years trying to explain this. His 1982 documentation was precise. Crown-of-thorns starfish can regenerate from a single arm, provided that a portion of the central disc remains attached. That phrase, *a portion of the central disc*, is the critical variable, not the whole disc. A portion. Here is what that meant for every culling dive. Gloved hands gripping a live animal under pressure. Arms splaying. Tissue tearing at stress points near the grip. Fragments dropped. Fragments caught on mesh edges and released without the diver noticing. Every improperly handled animal, not killed, not contained, but physically stressed and partially torn, was a potential multiplication event settling onto the sandy bottom below the reef crest. The program was not fighting the population. In some documented cases, it was seeding it. Janice Luff at the Australian Institute of Marine Science recorded 3 major outbreak waves between 1962 and 2000. The second ran from 1979 to 1981, arriving less than a decade after the first coordinated removal efforts ended. Correlation is not causation, but the timing did not go unnoticed. Every program since has confronted the same biological wall. The false answer, more divers, more funding, more hours underwater, collapses against one question nobody has cleanly answered. How small is the minimum viable fragment? And does science actually know? Here is the mechanism, and it changes everything. Acanthaster planci has no brain. Not a simplified one. Not a distributed one that approximates a brain. None. Its nervous system runs as 5 radial nerve cords, each extending down a separate arm, converging at a ring of neural tissue at the central disc. No hierarchy. No command center. Every arm already knows how to be the whole animal. That is not a metaphor. It is anatomy. Robert N. Dean at the University of Queensland documented it precisely in 1982. His finding, published in Marine Ecology Progress Series, crown-of-thorns starfish can regenerate from a single arm, provided that a portion of the central disc remains attached. That phrase, a portion of the central disc, is doing enormous work, not the whole disc. A fragment. Enough tissue where the radial nerve cords converge, and the arm carries a complete biological blueprint. Barker's 1979 work, followed by Ames-funded studies through the 1990s, put numbers to it: 6 to 12 months. That is the full regeneration timeline. One severed arm, bearing a sliver of a boral disc tissue, rebuilding itself into a complete, reproductively viable animal, spine by spine, arm by arm. Dione Dika at the University of Sydney confirmed this under controlled laboratory conditions in 2020. 6 tanks, 6 severed arms, each one regenerating on schedule. Which brings you to the injection programs. Ox bile delivered by syringe, directly into the body wall. The animal died. No cutting, no fragments, no accidental propagation. But here is what nobody expected. The culling teams were thorough. The numbers dropped in treated zones. 8 months later, the population in those same zones had recovered. Not partially, fully. The injections had not failed. Something else had happened, something upstream of the culling itself, something in the water, in the biology, that the intervention had not accounted for. So what was it? In 2012, the Australian Institute of Marine Science published the number that ended every argument. Between 1985 and 2012, coral cover on the Great Barrier Reef fell from 28% to 13%. 8%. Glenn Darth and his co-authors at Ames broke that decline into causes: cyclones, bleaching, and Acanthaster planci. Their conclusion, published in the Proceedings of the National Academy of Sciences: 42% of all coral lost. The single largest share was consumed by crown-of-thorns starfish. Not storms, not warming waters. Starfish. Morgan Pratchett at James Cook University confirmed it in 2014. Outbreaks caused more coral loss than bleaching events across that entire 27-year window. One species, 42%. Every culling program ever attempted killed individual animals. The divers with tongs, the injection crews with ox bile, the later vinegar trials beginning in 2014. None achieved a sustained population reduction at reef scale. Not one. The summary of every program trialed between 1962 and 2022 ends the same way. No intervention has demonstrated sustained population-level reduction at reef scale. Here is why the math never worked. One reproductively active female releases up to 60 million eggs in a single spawning season. 60 million. A researcher at the Australian Institute once wrote that number on a whiteboard, underlined it, and stepped back. The room went quiet. And then there is the arm. Go back to where this started. A severed arm resting on the sand. Fishermen believed they were solving the problem. They were not. Robert and Dean documented it in 1982. Provided a fragment of the central disc remained attached, that arm would regenerate a complete, reproductively viable animal within months. The reef lost 42% of its coral. The arm became a new animal. Both things happened simultaneously for decades, while programs ran and funding moved and reports were filed. The question science has not answered: at what population density does a crown-of-thorns outbreak become self-sustaining regardless of intervention? No study has established that threshold. No intervention has been tested above it. That question remains open, filed here, unresolved in the archive. The biology of one severed arm is still outpacing every answer we have found.